students, our today's topic is anatomy of dicot, anatomy of dicot and monocot root. We know in case of anatomy, we have to discuss internal structure. Internal structure. It means that internal structure of root, dicot root and internal structure of monocot root. At first, we will try to see how two types of roots differ from each other on the basis of their internal structure. And first of all, we will take dicot roots. And I will draw the structure here. For example, like this. And here we have some modifications, or we can say some extensions like this and like this. And we know that this is, here we call it as epidermis. Epidermis. And we know that this epidermis is single layered. It is epidermis. So the structure is like this. For example, this layer is single layer. Single layer, and this single layered structure we call it as epidermis. We call it as epidermis. And these are flattened parenchyma cells. These are actually flattened parenchyma cells. Parenchyma cells, and these are single layer. Single layer. Then there are some extensions like this. These extensions, or we can say modifications, we can say these are called root hairs. And function of these root hairs is absorb water and minerals. Root hairs, their function is to absorb water and minerals. Then there are cells like this, which we call as parenchyma cells. And these parenchyma cells we call as, this region is called as cortex. It is like this. Parenchyma cells. Parenchyma cells. And we have, at first we have taken dicot root. Structure of dicot root. Structure of dicot root. This region of parenchyma cells is collectively called as cortex. Then after the cortex, there is a layer. We call this as endodermis. dermis. Like this. This is called endodermis. And after endodermis, there is a sidrib. We call here as Casparian sidrib. And this Casparian sidrib, what is this? Is Casparian sidrib? It is a waxy layer. It is a waxy layer which is composed of suberin. And also it is impermeable. That means it does not allow water to flow. Then after this Casparian sidrib, there is xylem and phloem. 4 vascular bundles. 4 xylem, phloem. Xylem and phloem. And these vascular bundles, and these vascular bundles are arranged radially. So we can say in case of Dicots, the arrangement of vascular bundles is conjoint. That is conjoint arrangement when xylem is towards pith and phloem towards center. In center there is pith. And xylem is towards pith. So we have xylem towards pith and phloem towards periphery. Then there are this pith is having some specialized cells. Then we know that these vascular bundles are radially arranged and they are having cambium, so we can say these are open vascular bundles. These are of open type vascular bundles. So then there is a structure of elongated cells. Some elongated cells which are present here like this. Some elongated cells. These elongated cells are collectively called as pericycle. We call them as pericycle. Then there is a tissue which is present between xylem and phloem. This tissue, we call it as conjunctive tissue. It means that it is that tissue which is present between xylem and phloem. Then there are cells which surround the pith. There are cells which surround the pith For example, these are like this. These cells are called as parenchyma cells. These are called as parenchyma cells. So we can say the cells which surround the pit from this side, these are called as parenchyma cells. Then from this side there are cells which we call as sclerenchyma cells. So from this side the cells which surround the pit below, from below side or sclerenchyma cells. Sclerenchyma cells. Then in this way we can make the structure of a dicot root. Now we know that first of all there is epidermis, and this epidermis, these are flat parenchyma cells, and it is also single layered, and it is also outermost layer. Then there we know there are some extensions, or we can say modifications, in case of roots, which we call as root hairs. Then below the epidermis are present, as I have shown you, are present collenchyma cells, and this region of parenchyma cells, this region of parenchyma cells is collectively called as cortex region, collectively. Now the whole region of parenchyma cells, we know it is cortex region, and upper cortex is endodermis. So in case of roots there is a waxy layer, which I have told you, this is casparan strip, and this casparan strip is made of suberin, which does not allow water to flow, and we can say it is impermeable. Then there is some elongated cells in the form of pericycle, which is collectively called elongated cells in roots, which are present in roots. Collectively we call them as pericycle. So we know that this I have shown you in structure. There are 4 vascular bundles arranged in a radial manner, and this type of arrangement of vascular bundles is called as open type vascular bundle, because these are having cambium. Now when in case of monocots there are more vascular bundles as compared to dicots. Now I have told you there is conjunctive tissue, and these are some tissues which are present in between xylem and phloem here, which I have shown in my diagram. Then there are some specialized cells which are present in the center, which we call as pith, and pith is the central region, or specialized cells present in the center is called pith. So I have told you the cells which surround the pith or parenchyma cells, and also the cells which surround the pith from this side, these are sclerenchyma cells. And now we can see what are the features of these dicot roots. Features of dicot roots. Features of these dicot roots. First of we will see dicot roots have, these are having large number of dicot roots, or having large number of root hairs, and which I have shown you in the diagram that these root hairs absorb water and minerals, and these are also, I have shown in diagram that these are in the form of modifications which we call as root hairs. Second feature is that the number of vascular bundles is whole. The number of vascular bundles is whole as compared to monocots. So because there are 4 vascular bundles, I have told you, which are arranged radially, and these are arrangement of these cells is radial. So we can say in presence of that there is cambium. So we call them, and they are open vascular bundles. Now at third, as compared to monocot roots. Now the third feature is that there are open type of vascular bundles. There are open type of vascular bundles because they are having cambium, because they have cambium. Now the fourth one is that arrangement of vascular bundles is radial. Arrangement or vascular bundles are radially arranged. Vascular bundles are radially arranged. Now fifth is that all the cells that include pericycle, the cells that include pericycle I will write here this. The cells, fifth one, that include pericycle peak and vascular bundles, pericycle peak and vascular bundles. Collectively, they are called steal. Collectively, these are collectively it is called steal. It is called steal. S-T-E-L-E. Now we know that in case of when we take monocot root, we will see what are the features which differ a dicot root from a monocot root. Some features will be there will be some same features, or some features will be the same and some will be different. Let us try to know what are the features which resemble with monocot root. For example, it is monocot root. First of all, we know that there is less number of root hairs. So, for example, this is epidermis and there is less number of root hairs. Less number of root hairs. So we can say there is less number of root hairs. And you know these root hairs or the modifications or their some extensions which are present on the surface of a root. And second feature is that there is cambium, there will be no secondary growth. There is no having secondary growth. So we know there is not cambium because they have not cambium, because they have no cambium. So because of cambium, they are not having they are not undergo secondary growth. So we know that arrangement of vascular bundles is of vascular bundles is of closed type, So they are called closed type vascular bundles. Then here we know this epidermis is also single layered and it is also the outermost layer like this. And again there are parenchyma cells. Again there are parenchyma cells. And again this region is cortex region and parenchyma cells. Now there is also endodermis, which is the innermost layer of flower. There is also endodermis. And now the third feature is that there are also vascular bundles. Their vascular bundles can be in the 6 or more. 1, 2, 3, 4, 5, 6. Let us keep 7. So number of vascular bundles in case of monocots will be number of vascular bundles in case of monocots will be 6 or more. And we know these are closed type vascular bundles. So the arrangement of vascular bundles is a closed type, and this closed type arrangement of vascular bundles, it is called this condition is called polyurge. This condition is called polyurge. Now there is also pith. In case of dicots, there was not a well developed pith, but in case of monocots, this pith is very large. It is large and well developed. And there is also conjoint arrangement. Xylem is towards pith and phloem. Xylem towards pith and phloem towards periphery. So it was all about dicot and monocot roots. Thank you very much.